What if you could compare a new drug not to real patients in a control group, but to a virtual one? That is exactly what a synthetic control arm is. Welcome to the HealthTech Explorers channel. Imagine this. You are developing a new drug, but a classic randomized trial is difficult to run. Sometimes there are too few patients. Sometimes the disease is rare. Sometimes giving a placebo simply isn't ethical. And that is where the idea of a synthetic control arm comes in. In simple terms, this is a control group that is not recruited from scratch, but built from existing data. Researchers use electronic medical records, data from previous studies, patient registries, and sometimes even insurance databases. Then, with statistics and sometimes machine learning, they assemble a group that is as similar as possible to the participants in the current study. They look at everything that really matters: age, sex, disease stage, comorbidities, laboratory results. The goal is not to find a group that is just roughly similar, but one that is as close as possible to the real thing. Then, the comparison is straightforward. Patients receiving the new treatment are compared not with a conventional control group, but with this synthetic one. In other words, researchers are asking, what would have happened to these patients if they had not received the new treatment? And that is where the strength of the approach lies. It can save time and money because there is no need to recruit and wait for a separate control group. That is especially valuable in rare diseases, where every patient truly matters. And there is another benefit. In some situations, this approach is simply more ethical because it does not require leaving people without a potentially useful treatment. But there are risks too. The most obvious one is bias. Even if the synthetic group looks very similar, there may still be important differences inside it that we did not notice or could not measure. For example, access to treatment, patterns of follow-up, or the quality of care, and those factors can have a major impact on the outcome. There is also the issue of data quality. If the source data are incomplete, fragmented, or just messy, the synthetic control arm will be weak as well. And that puts the study conclusions at risk. That is why regulators such as the FDA and EMA approach this method cautiously. And that makes sense. Synthetic control arms are not used everywhere, and they are not meant to replace everything. Most often, they are used in oncology, in rare diseases, or as an additional tool when the classic study design does not work well or does not work at all. With the growth of artificial intelligence, this approach is becoming even more interesting. Algorithms can help find similar patients more accurately and detect hidden patterns in the data. But there is an important catch. The more complex the model, the harder it is to explain why it selected those specific patients. And in clinical research, explainability is not just a nice bonus, it is a necessity. So a synthetic control arm is not a magical replacement for traditional trials. It is more of a practical tool that helps in situations where the usual design is too expensive, too slow, or simply impossible. And perhaps the main question is no longer whether this approach will replace traditional trials. The real question is how carefully and honestly we will use it. Here at HealthTech Explorers, we explore complex topics in simple language. Together, we look into the subject and see how it works in practice.